Hello everyone, and welcome to the 32nd episode of the podcast. Today, I'll be discussing a group of organisms called Protists. Protists are a hugely diverse paraphyletic group, and they're defined as every other eukaryote that isn't a plant, animal, or fungus. You see, the plants, animals, and funguses are their own kingdom, but they're just a small slice of the world's total eukaryotic species. So all of those other eukaryotes, those are the Protists. In the previous episode, I discussed the two major domains, bacteria and Archaea. It's believed that at some point in the distant past, an archaeal cell absorbed a small bacterium, like it would have if it was trying to eat it. But instead of digesting it, the archaeal cell engaged in a mutual endosymbiotic relationship with the bacteria. The bacteria produced ATP for chemical energy, which the archaeal cell could use to power its own functions and actions and stuff. In return, the archaeal cell gave the bacterium chemical food and physical protection from the wilderness of open water. This symbiotic relationship created the eukaryotic domain, a domain that would see the emergence of virtually all complex macroscopic life. The domain eukaryote would evolve into complex microbial forms, into the first sexually reproducing organisms, into molds and algae and diatoms. All of these eukaryotic species and many more are the Protists. But among the domain eukaryotes, among the Protists, there are three very specific exceptions. Three exceptions that aren't Protists. And this is what makes it a paraphyletic group, as opposed to a monophyletic group. Within the eukaryotic domain, there are 7 phyla. 2 of these phyla are called the Opisthokonta and the Plantaea. The phylum Plantaea, just by its name, is a dead giveaway for the first exception. The phylum contains several protist algae, but it also includes an evolutionary lineage called the land plants, or the plants that grow on land. So there's the first exception, because those land plants are their own kingdom called land plants, or green plants if you want to include the green algae. And for all intents and purposes, the land plants aren't protists, so they're the first exception. Now, in the phylum Opisthokonta, there are 3 branches, or 3 lineages. The first is called the choanoflagellates, the second is the fungi, and the third is the animals. The choanoflagellates are protists, and they're closely related to the fungi. The fungi themselves are their own kingdom, as are the animals, who are also, closely related to the fungi. So the animals and the fungi are the second and third exceptions. So besides them, besides the land plants, the fungi, and the animals, every eukaryote is a protist. Because of these exceptions, the Protists are a paraphyletic group. They include a common ancestor, but they don't include all of the descendants. So this means that it isn't monophyletic, because monophyletic clades are those which include the common ancestor and all of its descendants. Because plants, fungi, and animals are descendants from the Protists, but they themselves aren't Protists, Protists as a whole are considered a paraphyletic group. Okay, so let me move on before I get all tangled up on definitions. This means that there isn't one single trait or characteristic that unifies all Protists. Most are single-celled, but others are large, growing into macroscopic structures comparable in size to land plants. The Protists are quite literally a biological bridge, a bridge through time, connecting the microscopic world with that of the larger animals and plants and funguses. Among the microscopic and macroscopic Protist species, there are vast differences morphologically, reproductively, and chemically. Because of these differences, the Protists are organized into phyla based on one or more traits shared by a group of genetically related species. These shared traits among groups of genetic cousins are called synapomorphies. They're defining morphological or genetic characteristics that help biologists group the Protists accurately. Scientists are increasingly using genetic testing to determine relatedness with other species. The greater the similarity in the genetic code, the closer the relation. These efforts have cleared up many areas of confusion by identifying 7 major classes or lineages within 2 major groups. The 2 groups, first and foremost, are called Biconta and the Uniconta, but I'll explain this in a moment. For now, let me start with the 7 major lineages. Since I already mentioned plants and the lineage Plantaea, I'll start with that one. So Plantaea, or Archaea plastida, has 4 evolutionary branches, called the land plants, the red algae, the green algae, and the Glycophyte algae. The land plants are the most recently emerged of these branches, and they're the Plantae that we're all familiar with because we eat them and we're surrounded by them all the time. They're trees, flowers, and grass and potatoes and stuff like that. The land plants diverged from the green algae, which share many similarities with the land plants like photosynthesis, but they exist mostly or almost entirely in or around water, like lakes or ponds or oceans. You might recognize algae as the greenish scum that forms on rocks near lakes and rivers. But did you know that many kinds of seaweed are also algae? Some microscopic algae are little more than floating blobs with two little flagella propelling them through the water. These diverse green algae are related to the red algae, which are equally diverse, including microbes, pond scums, and more species of seaweed. The differences between these groups are based mostly on minor genetic and biochemical differences. Red algae, for instance, are photosynthetic, just like green algae, but where green algae have a biomolecule called chlorophyll B, red algae have molecules called phycobiloproteins, which are pigments responsible for their red color. This takes us to the last group in Plantae, Glocca Fights, which are a small group of small organisms, much like microbial algae. The Glocca Fights are like a chemical bridge between bacteria and Archaea, and the other protists that connect them to Plantae. As the lineage Plantae branched off and diverged on its own, its cousin lineage, the lineage it left behind, would itself undergo divergences to produce 3 more classes of protist, each one bewildering in its number of species and its morphological diversity. These 3 lineages are the Rhizaria, the Alveolara, and the Stramenopiles. The Rhizaria are things like foraminiferans and actinopods, single-celled amoeboids characterized by the long, thin plasma tendrils that they use to move around. These thin, membrane-bound protrusions reach out from the main amoeboid body and they grab at food, or they wave and pull the Rhizaria through the water. Next are the Alveolara, which are creatures like the dinoflagellates and the ciliates, single-celled, linear-shaped microbes packed with tiny air pockets, or vesicles, that support their outer membrane. These flat little vesicle bags are a lot like the alveoli sacs in our lungs, which is why the lineage is called the Alveolara. The third lineage here is the Stramenopiles, which are much like the Plantae, except they're brownish instead of greenish. The Stramenopiles includes the water molds, the brown algae, and over 100,000 species of diatoms. The brown algae in particular evolved much like red and green algae, with species ranging from microscopic to shoreline vegetation to massive towering forests of kelp and seaweed hanging out in the ocean. So these 4 lineages, the Rhizaria, the Alveolara, the Stramenopiles, and the Plantae, are all part of a group that diverged from a population known as the Excavata. These are the oldest creatures with flagella, which is a synapomorphy shared by many other microscopic species in the lineages I already described. Most excavates don't have mitochondria, but some do. Some have 2 flagella, others have 4. Some have a mouth groove for specialized food intake, and some even have a rugged pseudostructure made out of support proteins, like a super primitive microscopic skull or an exoskeleton. The Excavata plus the other 4 are all part of a huge group called the Biconta, which are eukaryotes believed to share a common ancestor that possessed 2 flagella. So that's like the shared defining characteristic of this group. As the original Biconta diversified and formed many hundreds and hundreds of thousands of different species, some of them lost their flagella while others grew more. The Biconta diverged from the Uniconta, or the Amorphea, which themselves largely have a single flagellum or no flagellum at all. The 2 lineages in the Euconta added to the Biconta brings us to our total 7 protist lineages. These 2 Uniconta branches are the Amoeba Zoa, which are basically amoebas, and the Opistheconta, which you might remember includes the fungi, the animals, and the little protists called the choanoflagellates. All of these organisms share a specific genetic synapomorphy that strongly supports the claim of their shared ancestry. The genetic trait is a series of 3 genes, fused together and thus expressed as a group into 3 similar enzymes. All of these organisms share a specific genetic synapomorphy that strongly supports the claim of their shared ancestry. The genetic trait is a series of 3 genes, fused together and thus expressed as a group. When they're expressed, they form 3 similar enzymes, all involved in the production of various pyrimidine nucleotides, like dihydroortase and aspartate carbamoyl transferase. These chemicals are all called pyrimidines, which are the same class of molecule as the cytosine and thymine in your DNA. Humans are animals, and the animals are part of the Opistheconta, as are all of the funguses, and creatures called the choanoflagellates. I'll discuss the animals and the funguses, as well as the land plants and green algae, all in greater detail in future episodes. So for now I'm going to stick with the protists, in this case the choanoflagellates. These choanoflagellates are simple, single-celled organisms, possessing a single flagellum with a hood or a funnel that protrudes from the membrane to encircle the base of the flagellum stalk. As the flagellum spins and propels the choanoflagellate through the water, food particles build up on the funnel and get consumed. These choanoflagellates represent the genetic link between animals and fungus, having diverged from the animal lineage long after the ancestral population diverged from the ancestors of fungus. Next we have the Amoeba Zoa, which are themselves about 2,400 species of microbes with a large cellular membrane and no cell wall. They come in irregular shapes, like paint splatters, and they come in a variety of sizes, with some being less than 20 micrometers long, while others are more than 800 micrometers in diameter. The amoebas are unicellular, although, like with many other protist species, they'll often group up into multicellular masses as part of a reproductive cycle. One of the most notorious amoeba in the world is Malaria, which infects human blood cells and uses their bodies as hosts for part of the reproductive cycle. The blood cell eventually bursts, and it spills out a bunch of these malaria spores, which infect more blood cells and collectively cause serious, often lethal damage. The reproductive cycle of all protists is pretty varied as the species are from one another. Because protists represent a range of life from unicellular to macroscopic, they share many qualities with each, and reproduction is one of those shared qualities. I mean this in an extremely general sense, very fundamental. Some protists reproduce asexually, like bacteria and archaea, and these produce haploid genetic clones, while other protists produce sexually, like animals, to produce diploid offspring with blended genes. In many cases, the protists have a mixed reproductive cycle with both asexual and sexual components. You might have heard this before because it's a really popular idea, but scientists generally think that sexual reproduction and its inherent capacity for creating genetic variety were a response to diseases. Populations of asexual species are all clones, so if a disease can infect one of them, it could, in theory, infect all of them and kill the entire population. But in populations of sexually reproducing species, the intermixing and swapping around of genetic material creates unique genetic variety. A sexually reproducing population has more genetic variety, and thus is more resistant to diseases, as the pathogens have to adapt to every individual and their unique immune system. As in order to infect something, the pathogens have to adapt to every individual and their unique individual immune systems. Anyways, most species of protist undergo a life cycle where it transitions between asexual and sexual reproduction, or between haploid and diploid cells. Most important to understand about protist reproduction is that it really does vary a lot between species. Some species have a diploid form that lasts only briefly before undergoing meiosis to produce haploid daughter cells. These haploid cells can happily undergo mitosis, reproducing clones asexually for generation after generation. It's only when 2 of these haploid cells fuse to fertilization that the diploid form temporarily emerges. Other protists are the opposite, existing mostly as diploids with only a brief stint as haploids. These protists begin with the fertilization of male and female gametes, or haploid cells produced by a mature diploid cell. The mature diploids in these species can reproduce asexually through mitosis for multiple generations, with the smallest daughter cells going through meiosis to create new gametes. Consider the protist responsible for malaria, which is a genus of amoeba called Plasmodium. The Plasmodium uses both humans and mosquitoes as hosts for its life cycles. Haploid Plasmodium in the mosquito's saliva are injected into the human during a mosquito bite. The injected haploid cells travel through the bloodstream, where they first infect the hepatocytes of the liver, or the liver cells. They steal nutrients and they reproduce asexually until they burst through the membrane and kill the hepatocyte. These haploid spores then go on to infect the red blood cells, so the disease goes from the liver to the blood. The blood cells suffer the same fate as the hepatocytes. Their bodies are host for the spores, which reproduce asexually within them into male and female gametes. When a mosquito bites the human again, these malaria gametes in the human's blood are captured by the mosquito. Its blood meal contains the gametes, which then fuse into diploid zygotes. Back inside the mosquito, the malaria zygote undergoes meiosis and then mitosis to produce haploid cells. These haploid cells are injected into the next human to suffer a bite from the infected mosquito, and the malaria spreads again. In other species of protists, the reproductive stages include multicellular forms, large structures that are visible to the naked eye, and this includes stuff like seaweed and kelps and various algae. When the haploid and diploid forms are multicellular, the protist is said to be going through alternation of generations, which is a fancy word to say that each generation alternates between haploid and diploid. The haploid form is called the Gametophyte, and the diploid form is called the Sporophyte. The Gametophytes reproduce asexually through mitosis, while the Sporophytes reproduce using meiosis to produce spores. These spores divide through mitosis to become the multicellular haploid form. Alright, so I know this sounds really complex because I'm just throwing a bunch of complex phenomena at you all at once, and I'm going back and forth between all these big complex words, so let me break it down into something a little more simple. Hopefully the concept should become more understandable as I explain the process in greater detail with real-world examples. Consider the Stramenopiles brown algae called the Ectocarpus saliculosus. In its sporophyte stage, the Ectocarpus saliculosus looks like a small brown plant with fluffy little branches instead of leaves. The branches are only a few cells thick, but they end in globule-like clusters of multiple cells undergoing meiosis. In these globules, or buds on the end of the branches, the cells divide into spores. The globule ruptures, and hundreds of haploid spores are released into the ocean water. These haploid spores float along through the water, minding their own business, before they start dividing through mitosis. This asexual division turns them into columns of cells, like a stalk, or a root, or a stem, and eventually this division forms the Gametophyte. The Gametophyte form of the Ectocarpus saliculosus looks just like the Sporophyte form, except instead of little globules at the end of the branches, the Gametophyte has a column-like structure. It looks like a little ear of corn, or an obelisk, all made of haploid cells reproducing asexually through mitosis. The column structures on the ends of their branches can be composed of male or female gametes, and so when the column dissolves into its constituent cells, these cells, the gametes, will reproduce sexually by fertilizing with an opposite-gendered gamete. This fertilization produces a zygote, which undergoes mitosis to produce the diploid sporophyte, and the cycle begins all over again. If you thought that was still a little complex, don't worry, I'll go over other examples. I'll try and simplify it a little bit more. So let's consider another brown algae genus called the Laminaria, which undergoes alternation of generations, but unlike the Ectocarpus, the Sporophyte and Gametophyte forms of the Laminaria don't look the same. The mature Sporophyte form looks like a flat green hand with too many fingers, reaching up from a palm on top of a thin stalk. This form is very plant-like, and it can be easily mistaken for a marine plant. Within the fingery growths of the Laminaria sporophyte, haploid spores are being produced through meiosis. These spores are released, and they reproduce asexually through mitosis to form the gametophytes. In this case, the male and female gametophytes are separate, tiny multicellular structures with clear phenotypic differences. The male gametophytes form larger branching structures. Sperm cells bud off from these branches and saturate the local water in search of female gametes. The female gametophytes are smaller, with some of their cells morphing into exposed egg cells. The haploid sperm fertilizes the haploid egg, and this creates a diploid zygote. And the zygote divides through mitosis to grow up into a mature sporophyte, and from here the cycle begins again. The sporophyte uses meiosis to produce spores, which grow into gametophytes that use mitosis to produce gametes. The engagement of both haploid and diploid reproductive cycles is a trait widespread in the protists. Many species form multicellular structures and thus undergo alternation of generations. Even among species that share similar reproductive cycles, they may spend more time in haploid stage or more time in diploid stage. They may have identical sporophyte and gametophyte forms, or those forms might look wildly different and be different sizes. There really is a huge amount of variety, which shouldn't be a surprise in a paraphyletic group with hundreds of thousands of species. With that in mind, it shouldn't be too surprising that there's a variety in the way protists get food as well. Many protists are like bacteria in that they produce their own food through photosynthesis, or they absorb it from the surrounding water or mineral substrate. Some of the protists need to physically ingest a solid mass of nutrients, which can be done through a variety of mechanisms like filtering the nutrients from the water, or by extending their cytoplasm around their prey to capture and drag them into their bodies where they get dissolved. This latter approach is called phagocytosis, and it's quite popular among the protists, particularly the amoeba. But even within any of the 7 major protist lineages, there can be vastly different feeding strategies. Some species just use one technique, but others use 2, or even all 3. Within a major lineage, there can be different techniques, even within the same subgroup, like the alveolate ciliates, which use phagocytosis or a form of photosynthesis through endosymbiosis. This kind of photosynthesis is really interesting. If you listen to the previous episode, you'll know that I described Cyanobacteria, which are a group of photosynthetic bacteria that use sunlight for energy and who expel molecular oxygen. Some of these cyanobacteria are believed to have been absorbed by other cells, and instead of getting digested, they were actually integrated into their host's biochemistry, as also happens with the energy-producing mitochondria. The cyanobacteria absorb light and produce sugars, and in return they get protection and nutrients from the host cell. This is much like plants and the chlorophyll in their cells, but chlorophyll is also found in the other biconta groups like the excavata, the rhizaria, the alveolara, and the Stramenopiles. Sometimes, the chlorophyll in these other species will be wrapped in a quadruple membrane, indicating that they got their chlorophyll by absorbing another cell that had already absorbed the chlorophyll molecule itself. Not to run off into the woods on a tangent, but you can think of it this way. You're a single-celled photosynthetic organism, and you have a single membrane. A larger cell comes by and eats you, wrapping its membrane around you and absorbing you into its body. As you get absorbed, the host's cellular membrane that's wrapping around you will butt off internally, with you, the little photosynthetic cell, inside that membrane bubble. So now you have 2 membranes immediately covering your body. You have your own membrane, plus the outer membrane of the cell that absorbed you. This is called a primary endosymbiosis event, and it's why, for example, mitochondria have 2 membrane layers. They have the outer layer, which is like a tubular capsule, and they have an inner layer, which is a heavily folded mess of cristae and enzymes involved in the oxidative phosphorylation process. Anyway, now assume that the cell you're in, the cell that absorbed you and wrapped its membrane around you, is itself consumed by a larger cell, by some kind of protist like a rhizaria or an alveolate. Your host cell gets wrapped in a membrane bud just like you did when you first got absorbed, and now there are 4 membranes covering you. Because you're the photosynthetic particle producing sugars, you're what these cells are trying to get and to use. So over time, the cell that originally ate you is degraded and carved away until all that's left is you, the chloroplast, buried under a quadruple membrane. This is called a secondary endosymbiosis event. In this way, one cell in the lineage of green algae first absorbed a chloroplast, and as this cell reproduced and more green algae cells with chloroplasts appeared, some of them were also absorbed into other species, like the Rhizaria chlorarachniophytes and the Alveolata dinoflagellates and the Stramenopiles diatoms and brown algae. Other groups absorbed chlorophyll or mitochondria through secondary endosymbiosis, but then lost them due to some intervening evolutionary factor. Okay, so where was I before all of this talk about chloroplasts and photosynthesis? Oh yeah, food sources. So, photosynthesis is a popular way to get food, but so is absorptive feeding. This is common among bacteria and archaea as well, and also fungi, as it's probably the most basic way to eat that exists. The cell or the organism simply sits on its food or in a solution with food particles floating around, and as it's just sitting there, it absorbs the food through its membrane. It takes very little energy, and your food pretty much just comes to you. A lot of the bacteria, archaea, and protists that live in the guts of various animals are all absorptive feeders that help break down the food you eat. Absorptive feeders are also decomposers, like fungi, as they thrive on the decaying corpses of larger organisms. The most visceral and animal-like feeding mechanism is just raw ingestion. This occurs in protists that don't have a cell wall, as the wall would prevent the necessary movements, to capture and consume food like that. So, to ingest food, the protist has to flex and extend a portion of its cytoskeleton, extending microtubules and various fibers that support its body into protruding stumps. These stumps are kind of like lips, or tentacles, and as they reach out, they wrap around and capture the food item or the prey that's about to become eaten. After enveloping the food, the protist engages it with various digestive molecules, and this breaks it down piece by piece. Other protists aren't so aggressive as to seek out and hunt down prey. Others are more stationary, but they use masses of cilia to bring food towards them. The cilia are little hair-like structures, and they can all beat in rhythm, or in some kind of coordination to create focused water currents. The rhythmic beating or paddling of the cilia against the water creates currents that bring food closer to the cell, where it can then be acquired and digested. The cilia can also be used for locomotion. By waving or rippling in a coordinated effort, the cilia can propel the protist through the water. Flagella are also used for locomotion, although these are much larger and more powerful than cilia. The flagella are spun like a boat's propeller, or beaten up and down like the tail of a fish, which allows the protist to move relatively quickly. These mechanical flapping motions of the cilia and flagella they're pretty neat, but they're not nearly as weird as Amoeboid Motion. This is a special kind of movement that only some kind of protists can perform. Once again, it can't be done by cells with a cell wall, as the cell wall reduces the required flexibility. The cell needs the flexible liquidity of its membrane in order to perform Amoeboid motion. So much like when the cell creates protrusions to reach out and capture food, it can also create protrusions that act like feet. The membrane bulges and stretches to extend a leg or a foot, or what's called a pseudopodia, like a false foot. And this extended leg finds purchase somewhere, and then it drags the rest of the cell behind it. The protist's body slides along in this creepy, liquid-like way, its membrane-bound tentacles reaching forward and then pulling the cell behind it before reaching out again. If this isn't cool or creepy enough for you, consider that the biomechanics of this amoeboid motion is really similar to the contraction of animal muscle tissue. Filaments are slid past one another to flex and create movement, and this allows the organism as a whole to interact with its environment to move around. Because protists are the bridge between macroscopic life like animals and microscopic life like bacteria, it follows that this amoeboid motion might be an evolutionary precursor to the biomolecules in animal muscle. This really doesn't seem like too crazy of an idea, especially when you remember that besides the choanoflagellates that are also in the Opistheconta lineage, the Amoeba Zoa are the closest protist relatives to the animals. Alright, I've spent a lot of time talking about the physiology and taxonomy and evolution of protists. I think it's important to spend a lot of time talking about that stuff because the protists are a hugely diverse group, and simple, overgeneralizing statements can't accurately describe them. But with all of that said, I want to take a step back and talk briefly about how protists integrate into the larger ecology of life. As I've mentioned several times, protists can often be pathogens, whether it be various plant or fungal diseases, or something like malaria that's killed more humans than any other cause or pathogen in our entire existence. Other protists, like Giardia, cause diarrhea. Toxoplasma gondii causes toxoplasmosis, and Trichomonas vaginalis causes trichomoniasis. I could go on and describe in detail other protist diseases and pathogens and the immense amount of human suffering that they can cause, but it would just be an exercise in morbidity. On a more positive note, protists are hugely important for marine ecologies. They help absorb CO2 through respiration and photosynthesis, creating large populations of microbes that are an important food source for larger organisms like fish and whales. These marine protists are called plankton. For example, the plankton eaten by whales consists of a vast cloud of diatoms floating through open water. Of course, protists are also eaten by other protists, which creates a dynamic, interconnected food web. When all of these protists and fish and whales and other organisms die, their bodies sink to the seafloor, where more protists, like various decomposers and scavengers, can come and eat their bodies. Despite the activity of the decomposers, far more protists die in a given 60 seconds than can ever be eaten or decomposed, which means that their microscopic corpses will accumulate on the ocean floor. The protists play an integral part of the carbon cycle by engaging in the food web at every level. The corpses of these protists are often rich in calcium carbonate, and the immense pressure of the ocean crushes this as it accumulates on the ocean floor. Over geologic periods of time, this pressure and crushing compresses the calcium carbonate into sedimentary rocks and the soft organic tissue into petroleum. Alright, so that's about all I have for you today. The protists are a hugely diverse group of life forms, with body sizes ranging from microscopic cells to massive forests of kelp and everything in between. They live in a variety of places, including the guts of animals, the soil, and of course in pond water and ocean water. They're a critically important food source for many other species, making them an integral part of the food chains of any marine habitat. The protists are the group of organisms that help bridge the gap between the microscopic bacteria and archaea and the macroscopic land plants, fungi, and animals, all of which I'll be covering in the upcoming episodes. I hope you enjoyed this episode, and I hope you learned something cool about protists. As always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.